Chapter 4, Genetics and Cellular Function. So we just discussed the structure of DNA, the molecular structure of DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. We'll talk about why they're called those two different things, and in particular, why and how they're different from each other in structure and also in function. So we'll talk about the nucleic acids in the nucleus, protein synthesis and secretion, DNA replication and the cell cycle, and chromosomes and heredity. So those are our 4 topics of genetics and cellular function. And this is a funny joke. This is how DNA molecules combine. They go to a bar and one of them says, "Hey, can I buy you an amino acid?" Okay, that's DNA jokes. Organization. Well, you often think of DNA as a chromosome. We talk about it as chromosomes. I think it's just easier because people understand immediately, oh yeah, chromosome, yeah, that's DNA. Whereas when it's in the cell and not condensed, it's actually chromatin. Yeah, that's the thread-like DNA. It's not coiled. Rather, it's a thread. So this part here is the thread-like portion. And it's like that in the cell until the cell starts to decide to replicate, which is the cell cycle. So all of our cells replicate. You have new cells. Well, you're a totally new person in about 10 years. So our cells are replicating, replacing our stomach cells, which disappear fairly quickly, 3 days or so. We have a new stomach lining of cells. Your skin, every few hours, really, you lose some skin cells. So you're replacing your skin quite frequently. Your bone, I think, is the least frequent, although your bone cells are new every 10 years. The only ones that don't replicate your cells, don't duplicate, are nerve cells in your cerebral cortex don't, and muscle cells. So your muscle cells, you don't necessarily replace them. You don't replace them. Your muscle cells get either larger through exercise or smaller through exercise, but you don't get more cells. Anyway, that's a bit of an aside. Okay, so what happens to chromatin when your cells are about to duplicate or divide? The chromatin condenses, and then it becomes a chromosome. But we always talk about DNA as chromosomes. Yeah, compacted DNA. So here it is down here, already replicated, 2 chromosomes attached by a centromere. Okay, let's see what you know. In the chat, how many chromosomes do humans have? Thank you, eggs, yeah. I've got 48, 23, 46. So yeah, there are 23 pairs. So 46 individuals. 23, 23, just going to erase that for a second, call them what they really are, homologous pairs. So 46 individual chromosomes, but they're in pairs because the pairs are homologous. In other words, they are similar but not exactly the same. Just like, I make my students do this when we're in class. I won't make you do this, but say you have, well, you have usually 2 pairs of shoes. These are homologous shoes. If I were in the classroom, I'd make everybody take off their shoes and hold them up so that you can see that they're very similar, but not exactly alike. One is left, one is right. One is more scuffed than the other because, for whatever reason, you put more weight on one leg than the other. So they're similar, but not exactly the same. They're homologous shoes. So our chromosomes are homologous. How many chromosomes does a fruit fly have? And I'm asking this about fruit flies because fruit flies are used for genetic research into human genes. As a matter of fact, David Suzuki, he did his degree in genetics, and he used fruit flies by the thousands, I would imagine. Yeah, we have fruit flies at work, and we use them also to do some genetic study. You probably don't know because fruit flies don't come up in conversation all the time, especially not the number of chromosomes that they have, but they have 8. They have 8. But interestingly, fruit flies have, I've heard varying numbers, but say they have about 20,000 genes, 8 chromosomes. Humans have about, the number changes sometimes, but around 22,000 genes. So only about 98% of human DNA is coding, codes for any proteins at all, as far as we know. Now, this is an interesting question. Butterflies, how many chromosomes do butterflies have? Well, I've looked into that, and there's lots of different answers, but there's many species of butterflies, and they can have anywhere from 10 to, get this, 360 chromosomes. That's an enormous number of chromosomes. There's no way that a butterfly needs that many chromosomes. But what happens sometimes is a process known as polyploidy. That's when chromosomes duplicate, but they don't divide into separate cells. So you end up having cells that have lots of chromosomes that have been duplicated. So if, for example, a chromosome during meiosis ends up 2 in one cell, so instead of having 1 from mom and 1 from dad, there's 2 from dad. And these cells combine, and for that particular chromosome, you've got 3. That's called triploidy. And that is the cause of Down syndrome. So that is something that occurs, is more likely to occur with individuals, I think it's 35 and older. The likelihood goes up to, I think it's 1 in 7,000. I'm not exactly sure, but the likelihood of triploidy does increase. But it can also occur at younger ages. So, wait. One thing I wanted to tell you about, which is very interesting, it's these proteins here. They're called histones. Together, these are called nucleosomes, but so this is also a histone. Some very fascinating recent research about histones, and that we've always thought, oh, it's only DNA that gives you traits. We've always said that for a long time, since Lamarck. And that was like in 1917. So when the Russian Revolution occurred, the Soviet Union took on Lamarckian genetics, which was that you acquired traits in your lifetime and then you passed them on to the next generation, which is different from Darwinian genetics or Mendelian genetics, whereby you pass on those traits via your DNA. And whatever you have, you have. But it looks like there is some traits that are passed down not through DNA, but rather through proteins like a histone that you also inherit along with the DNA. It's a very interesting field of study. It's called epigenetics, if you're interested in looking at that. Epigenetics. So let's look at a condensed chromosome. So whenever you see a chromosome or DNA in diagrams, almost always as a chromosome instead of a real long, instead of a very long thread, because that would be too cumbersome. So the question is, histones are found in chromosomes. Histones are found in chromatin. So the long thread-like DNA is wrapped around the histones. Histones are a protein. So that's a good question. Yeah, thank you. So this is a chromosome. I'm just going to familiarize you with a bit of terminology. So loci, which just means places, that is where you find the genes on the chromosome. And a gene is a sequence of nucleotides. Yes, so DNA is a long string of nucleotides and DNA is simply a sequence within that. And this is condensed DNA, and this singular is called a locus. Just means the place where that gene is, essentially. Yeah, so this is the centromere, and this is the location of a gene. This is the location of another gene. I'll just make this a bit larger. So genes are often just given 3 or 4 letters to indicate them. I don't know what all these letters mean in longhand, but yeah, these are all genes. And you might be wondering, how do we know where they are on the chromosome? And that's an interesting question. That's an area of genetics which utilizes a lot of cross fertilization, not cross fertilization, cross breeding. Sorry, cross breeding. Yeah, so you can figure it out by cross breeding, and a phenomenon that occurs during meiosis, which is called crossing over of chromosomes. So yeah, it's very interesting. So say, for example, you have, this is getting ahead of ourselves a little bit, but you have homologous chromosomes that during meiosis are lining up. Well, in the process of meiosis, oh, whoops, sorry. Wait. Scratch that one. That should be red. In the process of meiosis, there's been some crossing over. And so this chromosome now has a bit of the other homologous chromosome. So homologous chromosomes don't always stay the same. They just don't always stay the same because of this crossing over. And what's interesting is that if the gene is down here somewhere, it's more likely to get crossed over more often. If the gene is up here somewhere close to the centromere, it's probably not going to get crossed over very often. And so you can tell with lots of experimentation, lots of crossing over experiments, that this gene must be further toward the end of the chromosome than this one. And that's how you figure out where these loci are. So I'll write that down in a summary. This is crossing over of homologous chromosomes and cross-breeding experiments reveal loci of genes. And that's all I'm going to say about it at the moment. Okay. So here is the Y chromosome with its loci. There's the testes determining factor. There's the gadgetary gene, also known as the MAC locus. These are all male traits. There's the channel flipping gene, also known as the FLP locus. Okay, these are not real. Catching and throwing, the sports page. You know, this is, I got this off of somewhere. I don't know where. I haven't found one for the X chromosome yet, but if you ever find one, then you can let me know and send one in. Those are pretty much all made up except for the testes determining factor. Yeah. So if you wanted to know more about this experimentation, I encourage you to look into it. It's not something we will delve into in more detail at the moment, however. The Y chromosome. Here are our nucleotides. Adenine. What part of the nucleotide is adenine? Is it the nitrogenous base, the sugar, or the phosphate group? You can write that in the chat. The adenine, is it the nitrogenous base? Is it the sugar, or is it the phosphate group? Nitrogenous base, good. Nitrogenous base. Yeah, so this is our DNA nucleotide structure. This is our ribose, but it's not just any ribose, it's a deoxy ribose sugar. And it's called that because you'd have to have something right next to it to compare. But look, here we're missing the oxygen. Missing. That's why it's called deoxy. The oxygen is missing. Yeah, and we've got our phosphate as well. That's nucleotide structure. And DNA is simply a string of nucleotides. And the nucleotides, there's 4 of them. There's only 4. Guanine, cytosine, adenine, and thymine. Some of them you'll notice are double rings and some are single. A double ring always pairs with a single ring. And that's because that's just simply how these hydrogen bonds form. So between cytosine and guanine, there's a triple hydrogen bond. And between adenine and thymine, there's a double hydrogen bond. Yeah, so that's why they pair up that way. It's called complementary base pairing, and it's always the same. Cytosine with guanine and adenine with thymine. So here are these nitrogenous bases. So that's what's different between these nucleotides, the nitrogenous bases. The purines are double ringed. The purines are adenine and guanine. And the only way I can remember this is that purine has a U in it and double has a U in it. So the purines are double ringed. The pyrimidines, however, are not. They're single ringed. But what's interesting is that they all have a Y: pyrimidine, cytosine, and thymine for DNA. Well, that's one way you might use that to remember. Uracil has to get in there though, doesn't it? Uracil is part of RNA, not DNA. You get uracil instead of thymine. Thymine is part of DNA. So that's a difference between DNA and RNA, which we're exploring. So here's some complementary base pairing. A nitrogenous bases forming hydrogen bonds, base pairs ATCG. It's called the law of complementary base pairing, which is interesting because it makes it quite easy to replicate. All you need is one string, and you can just make another one based on complementary base pairing. So the function of DNA is to code for protein synthesis. A gene is a sequence of DNA nucleotides. It codes for one, and we used to think, you know, in the old days when I went to university, we used to think that there was one gene, one protein, or one gene, one enzyme. That was the hypothesis. You could always only get one gene or one enzyme from a gene, but that turns out not to be true. And that's one of the reasons why a fruit fly, which has the same number of genes, and a human can have a much more complex structure, because you can have more than one protein per gene. A genome is all the genes of one person. We have about 22,000, 98% non-coding. So this is the central dogma of biology. DNA replicates. Sorry? Was that a question? Okay. DNA replicates, makes copies of itself, and DNA is transcribed into RNA, and RNA is translated into a protein. We'll go over all those functions, but we're going to start by looking at RNA. RNA is not double-stranded as DNA is, it's single-stranded. It's single-stranded, and it is considerably smaller. These are some various viruses. Viruses contain often what's known as a positive strand RNA. The important thing, though, is that it's an RNA strand normally, not a DNA strand. The genetic material of the coronavirus is an RNA strand. But when RNA from the virus enters our cells just go, oh, here's some RNA. I guess I'll translate it into a protein. So that's how viruses use your cell in translation. So all of our processes are controlled by DNA. So this is an interstitial cell of a testis. The interstitial cells. And the interstitial cells are responsible for synthesizing testosterone from cholesterol. So here's cholesterol. It readily goes into the cell because it's a lipid, but it gets converted into testosterone in the cell. And the reason is that DNA, which is transcribed into messenger RNA, which is translated into this enzyme, gets activated. It gets activated by a signal from elsewhere. In this case, the signal is ISH. That's also known as LH, luteinizing hormone. That comes from the pituitary. The pituitary signals the interstitial cells to start making testosterone. If there's a lot of testosterone, the pituitary will be turned off. So it's a feedback system. So DNA is really responsible for the synthesis of all cell proteins, either directly or indirectly. Or indirectly when one molecule to another. It can't do it without the enzyme, and the enzyme is encoded by DNA. So I'm just going to do a couple more things, and then I'll give you an exercise to do. So here's a preview of protein synthesis. DNA, always in the nucleus, protected in the nucleus. Transcription. So one DNA strand is used to make an RNA strand. Only one of the DNA strands is used. It's modified. That RNA is modified. Now we have a mature messenger RNA goes outside the cell through a pore, and outside the cell is translated into a protein. So that's the function we're going to look at. Amino acids, there are 20. Some of them are essential, 11, I think. And you need some of those in your diet. You can't synthesize them. An essential protein or vitamin is something that you can't synthesize, you need from your diet. So interestingly, the genetic code is based on 3 nucleotides in a row on the messenger RNA. So messenger RNA, say it's something like this, GCA UGC. Each triplet is known as a codon. And each codon codes for an amino acid. So if I were to use this first codon, GCA, which amino acid is that coding for? Can you figure it out by looking at this table? This is the first base, the second base, and the third base. Try to figure it out using the table. There's only the first 3 letters are given. That's good enough. Yes, alanine. ALA stands for alanine. Yeah, so this is the messenger RNA, and this is important to remember. The code, in this case, the code is taken from the messenger RNA. It's not any other NA. It's not DNA, it's not T messenger RNA, or tRNA rather. It's always messenger RNA. That holds the code the way that we're using it. Good, so you take the first base, G, and find it here. Then you take the second one, C, and find it here in the column. So you take this column here and find the amino acid in the corresponding row. And then the third base is over here, A, which is on this row. So that's how you find the amino acid. And we'll do an exercise using that in a moment. The first stage is transcription in protein synthesis. Transcription, making an RNA strand from DNA. So this is DNA, and this is RNA, messenger RNA. But this messenger RNA goes through some more processing called alternative splicing. So here, transcriptions happen here, and you've ended up with this strand. Each strand of messenger RNA, when it's initially transcribed, has sections that we call introns and exons. And interestingly, the introns get spliced out. Yeah, some of them are spliced out. But that could result in different sequences of messenger RNA in the end. So here's messenger RNA 1. It's just simply the A, the C, and the D. Everything else has been spliced out. This is very simple, of course. There's going to be way more. Messenger RNA 2, different splicing. Different splicing. You end up with exon B, D, and E. And the rest has been spliced out. Splicing means to cut, by the way, to cut or to split. Yeah, so when they go through translation, now you've got 3 different kinds of proteins. So that lends itself to the idea that you can get a complex organism like a human from only 22,000 genes. And translation occurs in the cytoplasm. That is where the messenger RNA is used to make a protein. It's basically red. So here's your messenger RNA, this long string here. And it attaches onto your ribosomes. This is the large subunit, the small subunit. And each amino acid, so that's an amino acid, is attached to a transfer RNA, and we'll look at that closely in a moment. But this transfer RNA has 3 little nucleotides that are anticodons. And the anticodon matches the codon on the messenger RNA. So we'll look at that more closely. We're actually going to look at a video about that as well. Transfer RNA is very interesting. It's a molecule of RNA, and it has an anticodon on one loop. And on the other end, it has an amino acid accepting end. So only this transfer RNA will have this amino acid. With a specific anticodon will carry the specific amino acid. It requires energy. It does require ATP. So here is the sequence of events. We'll also watch a video on this. Number 1, messenger RNA has left the nucleus. Ladies and gentlemen, messenger RNA has left the nucleus. You'd understand that if you knew anything about Elvis Presley. So you might not even know who Elvis Presley is, but anyway, he was a famous singer, the king of rock and roll. Anyway, he would have so many fans when he was doing his concerts, and they'd all want to see him. And eventually the announcer would have to say, ladies and gentlemen, Mr. Presley has left the building, so they wouldn't crowd up on the stage. So messenger RNA comes out, it binds to a ribosome, or rather the ribosome binds to the messenger RNA. And then at the same time, transfer RNA is binding to an amino acid. And then the transfer RNA anticodon binds here with its amino acid. And this is going on an ongoing basis. There's already a tRNA here. It transfers its entire string of amino acids to the newbie. Transfers it to the newbie, and then this one leaves. Goes away to pick up new amino acids. Yeah, big party. So yeah, then when it's all over, the ribosomes break apart. Translation. And well, you know, this makes it look like, oh, it's one messenger RNA read by one ribosome and you get one protein, but that's really not how it works. Rather, there are many ribosomes reading the same messenger RNA. So many ribosomes read the same strand of messenger RNA, resulting in many proteins, many hundreds of proteins. So this is showing here, this is a electron microscope micrograph. Those are all ribosomes, and these are amino acids being built. Or I should say being attached to make a protein. So here's a quick review, and then we'll do our exercise. So starting with DNA, double helix. Secondly, one strand of DNA is chosen. It's chosen by the enzyme that's responsible for this process, RNA polymerase. Pre-messenger RNA is made, spliced. The mature messenger RNA is here. These are the codons. There are some codons that code for start. It tells the ribosome, this is where you start. And some for stop, telling the ribosome, this is where you stop. The anticodons are here of transfer RNA, here at the end. They are attached to very specific amino acids, which at the end of the process form a peptide. A peptide is a string of amino acids. A polypeptide is a long string of amino acids, and a protein is a really long string of amino acids. I'd like to stop there and start the exercise. All right, thank you for watching.